In my group, we try to understand the molecular basis of plant response to stress, as this information can be used to improve plant performance under such situation. So we know this information, and we believe that this knowledge would contribute to ameliorate the consequences of global warming and to feed the increasing world population. So among other problems, we study the mechanism by which plants handle water for growing. So at plant level, major water losses occur through transpiration at leaves. There are these tiny pores, which are called stomata, which are open during the day and closed during the night, that participate in gas exchange for photosynthesis. So there is where the carbon dioxide comes into the plant in order to fix these carbon atoms into carbohydrates. And the point is that as they are covered by water vapor, for the net balance is that for each molecule of carbon dioxide that comes into the plant, there are 100 of water molecules that go to the atmosphere. So we wonder if it would be possible to optimize this balance to reduce water consumption in the case of a stress event, or if we could improve crop productivity by promoting stomata opening and consequently enhancing photosynthesis if water is not a limiting step. So we first study how this works at molecular level in planta. So it is well known that behind all this opening and closing of these stomata, there is this tiny molecule, tiny but rather complicated abscisic acid molecule, that is regulating this opening and closing. So in a stress event, the ABA concentration in the cytosol increases and the stomata is closed. When the stress is released, the concentration of ABA diminishes and the stomata opens back. It is also known that there is a group of small molecules, which are called ABA receptors or pills, that are regulating the function and the activity of a group of phosphatases and kinases that in turn regulate by phosphorylation ion channels and transporters that trigger water fluxes that drives the stomata to an open or closed conditions. So this rather complicated pathway can be reduced by this chemical equilibrium. So in the resting state, the ABA receptor pills are in the EPO form, and when the ABA increases, this triggers the formation of a ternary complex between the ABA receptors, the ABA, and the phosphatases PP2C. This inhibitory complex releases the kinase SNARC2 in order to phosphorylate and activate these channels I already mentioned. So this triggers stomata close, and when the ABA is decreased, we move in this equilibrium, we shift this equilibrium to the left-hand side to open the stomata back. So this pathways is well characterized from the structural point of view. The ABA receptors are monomers or dimers that contains a large hidden cavity in which ABA binds. This binding triggers a structural reorganization of the ABA receptors, and this in turn make possible the interaction with the active site of the phosphatase, leading to this inhibitory complex that triggers the activation of the kinases. So if we want to control this, what we want is to unbalance the equilibrium in a target manner. So we may want to move or to balance the equilibrium to the right-hand side, to the open stomata, if there is, or if we seek for protection to draw stress, or we may want to improve the growing and then unbalance the equilibrium to the left-hand side in order to produce inhibition of the kinase SNARK2, and then open the stomata. Okay, so in order to do so, there are two approaches. The first one is the genetic approach that will involve altering the gene expression in order to produce an imbalance of this equilibrium. So on the top of the social perception of such approach, there is a major drawback of this approach. Is the fact that transgenic plants and genome editing plants lead to a constitutive activation of the stress-responsive pathway. That means that the plant would be permanently stressed, and this is something we don't like. Instead of this genetic approach, there is another approach, which is called the chemical approach, that seeks for and balance the equilibrium by the formation of the product by finding chemical compounds that triggers or by mimicking the effect of ABA, triggers the unbalanced equilibrium to the closest stomata. So instead, this chemical approach has major advantages. First one is does not require genetic manipulation. Second, could be used on demand. So only when it is required, we can add such compound and trigger the equilibrium or balance the equilibrium to one or to the other side. And as ABA is a universal pathway, can be used in a general way, even for autochthonous varieties. So for the small farmers that produce autochthonous varieties. So we wonder if it is possible to design such a compounds, such a ABA mimicking compounds, and supply them to crops as if they were an agrochemical. So to achieve this objective, we underwent a structural-based campaign for the discovery of such a new compounds. Here on the right-hand side of this slide, you may see the typical scheme showing the pipeline that starts with an in silico screen, followed by biochemical validation, structural validation. This is usually cycled with a chemical optimization step, which goes back to the biochemical validation in order to optimize the compounds, the very first compounds obtained by the silico screen. And then it moves back to the structural validation. What is different in plant is that as applying chemical treatments to plants can be tested in large population, is low cost, and raises no ethical concerns. So there is no animal testing, no anything at all. So it is possible to include the cycle of optimization in the cycle of optimization in vivo assays. So this makes the projects move very fast from the very first in silico screen to the in vivo assays, even in crops that could be tested in a large number of individuals. In any case, to achieve or to, as for any other structural-based campaigns, we need to get first realistic structural information. Second, we need to optimize the fast and reproducible ligand binding assays in order to produce the biochemical validation. We need to have available crystallization conditions that yield to crystals from moderate to high resolution to produce the structural validation. And of course, we need to define clear phenotypic assays in order to in vivo validation. So at the starting point of this scheme, the initial source of structural information came, in our case, for fragment screening. In fragment screening, we look for small chemical fragments that bind to the protein. We soak the crystals with these fragments and then use X-ray crystallography to see exactly how they bind, as it is being shown in this movie. Here you can see this produce a very rich information in order to succeed in these in silico campaigns. Here you may see the overlap of all the fragments that lie into the ABA binding pocket, and this makes possible to do a sort of molecular cartography of the binding sites. So we can realize that in this area on the top of this skin are mainly H1 receptors, the same as here, and there is an hydrophobic belt that is full of hydrophobic residues of our ligands. On the top of this, we got experimental information on the flexibility of the receptor. So the receptor is able to move in order to accommodate these ligands and this sort of breathing of the protein. This can be seen, as we've seen, at the level of folding, at the level of side chains, and this definitely affects the shape and the volume of the cavity or the binding cavity. So goal from CCDC allows or enables the implementation of this experimental information in a very efficient way. Kepa has mentioned something. Ensemble docking will account for the flexibility of the protein as well as the flexibility of specific residues, and it will be possible to do a pharmacophoric selection that accounts for the characteristic of the binding site. So we can say in this particular area, please scale or weight up all the disposals that contains hydrophobic residues or H-bond donors. So using this algorithm or this program, what we try is to seek for natural compounds mimicking ABA that were present in food. This was a very interesting approach because it would avoid contamination risk, bypasses all the regulatory constraints because they are natural products that are present in food, and focus on safe and food-derived compounds, and definitely cuts costs and speeds up the discovery. To do so, we use Phenol Explorer database. We create this database consisting of 500 polyphenols, and we found that the hydroxycinnamic acids were predicted to bind ABA binding pocket. It was very interesting because these acids are very abundant in coffee residues. Coffee is the most popular drink in the world after the water, and then it's one of the world's major organic wastes. So this was really promising to get this sort of compounds as ABA mimicking compounds. So in the biochemical validation, we realized that they display antagonist activity. That means that they could be used for improved growing. So they unbalance the equilibrium to the open stomata, not for the closing stomata. So this was quite interesting, and structural validation provided a rationale for that, as we saw that they mimicked the interaction of the ABA into the binding pocket, but then they extend out of the binding pocket, thereby blocking the phosphatase binding. So at this point, what if we try to improve the efficiency of these compounds by enlarging further the molecule out of the active site. So we found that this molecule, anecdote of coumaric and tyrosine, were predicted to bind the open form of the receptor and display also a high score in the docking simulation. So we underwent what's so-called scaffold merging campaign, in which we seek to merge these three hydroxycinnamic acid with aromatic natural tyrosine and phenylalanine aromatics, and we found that this compound larger was able to increase the activity of these compounds by at least 1 order of magnitude in this ligand binding assay. And we, by solving this structure, we realized that there were, as the other residue, expanding out farther of the active site, stabilizing an open form of the receptor and blocking the formation of the inhibitory complex. So that was very interesting, and we, of course, end up the project by doing some in vivo assays and checking that in vivo germination and other ABA-dependent phenotypes were definitely blocked by the action of these hydroxycinnamic acid derivatives, and that they were active. Just to end up with this presentation, just illustrate the groups involved in this project. A group in Valencia, where the plant experiments were carried out. This is essential because it's central for the pipeline discovery of drugs to plants. My group in Madrid, which was focused on the structure data and biochemistry, and Robert Sirac at the Cambridge Crystallographic Data Center, that was helping us and teaching us how to use all the tools that are required to develop this project. And thank you very much for your attention.